Bismillahirrahmanirrahim. The mechanism of insulin secretion is discussed in this video. Glucose is the main regulator of the insulin secretion. Beta cells have a glucose transporter that allow glucose influx proportional to the blood glucose concentration. And once inside the cell, the glucose is phosphorylated into glucose 6 phosphate by glucokinase, the rate limiting step for the glucose metabolism in the beta cell. This step acts as a glucose sensor, adjusting insulin secretion based on glucose level. Then there is this glucose 6 phosphate is oxidized, and after oxidation there is production of ATP, adenosine triphosphate. This increased ATP synthesis inhibit ATP sensitive potassium channel that are present on the beta cell membrane, and closure of these potassium channel will result in depolarization of the membrane, and after the depolarization of the membrane there is opening of voltage gated calcium channels, and the calcium influx that is resulted due to this opening of these channels triggers infusion of insulin containing vesicles within the cell. These insulin containing vesicles that are present just below the cell membrane, they are released into the extracellular fluid or blood by the exocytosis, by the process of exocytosis. So the other factors that affect insulin secretions are stimulatory nutrients, certain amino acids that increase intracellular ATP, enhancing insulin secretion, then enhancing hormones such as incretins, glucagon, GIP or glucose dependent insulinotropic peptide, and acetylcholine. They raise the intracellular calcium uh, through other pathways, amplifying the effect of the glucose. Then there are some inhibitory hormones such as somatostatin and norepinephrine, that is alpha adrenergic activation. They inhibit insulin exocytosis. Uh, Sulfonylurea that are used in type 2 diabetes, they stimulate insulin secretion by blocking ATP sensitive potassium channel, mimicking the ATP effect. So the insulin secretion is a multi-step process. The glucose has entered, it is converted into glucose 6 phosphate via glucokinase, then ATP increases. This increase in ATP will result in depolarization of the calcium channel. And then calcium influx is there, and insulin vesicle, they secrete the insulin by the process of exocytosis, and this process is modulated by nutrient, hormone and drugs. Just to revise this process again, so here is the pancreatic beta cell, and this pancreatic beta cell, in response to the increased glucose levels, there is expression of the GLUT2 on the beta cell, and in response to this increased glucose, there is increased entry of the beta cell via the GLUT2 transporter, this yellow channel, which is sensitive to blood glucose concentration. The glucose is phosphorylated inside the cell by the enzyme glucokinase. This phosphorylation is in the form or results in the form of glucose 6 phosphate. and glucose 6 phosphate is oxidized through cellular metabolism, and there is production of ATP, increased ATP. Then these increased ATP closes the ATP sensitive potassium channels that are present on the membrane, and this closure of these channels result in the process of depolarization of the cell membrane, and depolarization of the cell membrane opens up calcium channels. These are the voltage gated calcium channels, and calcium ions, they enter into the cell, triggering the next step. And there is the next step is the stimulation of these insulin containing vesicles to move and to fuse with the cell membrane, and then insulin is secreted into the extracellular fluid and then into the blood stream by the process of exocytosis. So this process shows how blood glucose levels are directly linked to insulin secretion, with ATP and ion channels acting as a cell mediators. So this table is listing the factors that influence insulin secretion, divided into two categories: factors that increase and those that decrease insulin secretion. Factors that increase the insulin secretion are increased blood glucose, increased free fatty acid, increased blood amino acid, gastrointestinal hormones such as gastrin, cholecystokinin, secretin, gastric inhibitory peptide. Then there are other hormones like glucagon, growth hormone, and cortisol. Parasympathetic stimulation will result in secretion of acetylcholine, and it also increases the insulin secretion. Then beta adrenergic stimulation, insulin resistance, obesity, and sulfonylurea drug, glyburide and tolbutamide, they are also responsible for increase in secretion of the insulin. While the factors that decrease the insulin secretion are decreased blood glucose, fasting, somatostatin, alpha adrenergic activity, and leptin. All these are responsible for the decrease in insulin secretion. So this table is showing how nutrients, hormone, autonomic stimulation, and drug can either stimulate or inhibit insulin secretion, highlighting a complex regulation of insulin in the body. Thank you for watching this video. If you have any query, please write in the comment box.